okay. Now, some characteristics can be transferred to the offspring in relation to just being just one characteristic of the organism. Like, for example, just the characteristic of tallness can be transferred from a chromosome in that working on its own. When we are looking at just one one trait of the organism that is to be transferred in terms of the genes from the parent to the offspring, we call that monohybrid inheritance. Alleles for a trait will separate during production of gametes, which will contain one of the alleles of the trait that can be the same or different. These gametes fuse during fertilization and become two alleles, and the trait which may be different from those of the other parents with segregation and recombination yielding a genetic variation. So when we cross one parent to another parent, the dominant trait will be seen on the phenotype. And that is the reason why we need to look at monohybrid inheritance. The resultant generation is called the filial generation or the first generation. And if we cross them, the children again, we call them the F2 or filial 2 or a second generation. Right. When we're doing monohybrid crossing now, we are just going to look at a certain characteristic of the organism and see the interaction of the alleles as they are going to occur at the end of the crossing. For example, we are going to show the impact of crossing peas with a specific allele coding for a rounded pea seed and another allele, and in this allele we're having two extremes. Either we are going to have a rounded pea seed that is represented by capital letter R, or a wrinkled pea seed that is represented by small letter r, and these are recessive alleles. So how are we going to do this? So we have this genetic cross that we are seeing here. This genetic cross is going to show us how monohybrid crossing is going to occur. So we have these two parents here, which are presenting a phenotype of the female having the rounded pea seed, and then the male represented by a wrinkled pea seed. From here, what is coding for these phenotypes are these pair of alleles. So this is homozygous dominant for the female, and then homozygous recessive in the male. If they are then separated through meiosis to form gametes, these gametes will be found in the individual chromosomes that are found in the gametes. So they may exist in the individual capacity, with this allele for dominance existing on its own as a gamete, and this one also segregating and existing its own in the gamete. Also, this is going to occur in the male, like what is occurring in the female. So the chances of them combining at fertilization are 50-50, such that this first gamete can cross with this first gamete from the male to produce a combination with capital R, small letter r, which is heterozygous, or this first gamete will cross with this gamete so that it can produce this pair of heterozygous R and R. Similarly, it will occur with this gamete fertilizing with this gamete to give us a heterozygous outcome. Also, this gamete will combine with this gamete to give us a heterozygous outcome. So the F1 genotype is all the result in offspring carrying an heterozygous combination or a heterozygous genotype, so that all the seeds that are going to be produced here have a phenotype of of being a rounded seed in their first generation. That is what we need to look at monohybrid crossing. We also investigate that when we're looking at codominance. So codominance appreciates that some alleles are dominant to others. But what if for a specific characteristics, it is two alternative alleles that are both dominant? And so the resultant outcome or phenotype is a blended outcome, so that both the characteristics of the dominant alleles are present in the resultant phenotype. We call this codominance. That is when we see things like a white flower crossing with a red flower to produce a different type of flower that is blending the characteristics of the gene for white flower and red flower that can become as pink. This is a genetic cross that can resemble what we are talking about. If the parental phenotype is the female having a red flower and the male having a white flower phenotype, then we look at the genotype having this homozygous dominant for red flower and also this one having a homozygous dominant for white flower. If these then segregate during fertilization, the gametes will carry one of the alleles in the genotype, homozygous dominant genotype. As such, we are going to see this gamete either combining with this gamete to give us a heterozygous genotype in the F1 generation, and if we continue crossing them, they will give us the same outcome of heterozygous genotype. But now the two alleles are dominant, and because they are dominant, we are going to see a blending of the characteristics such that the resulting phenotype in the first generation is going to be all pink. It does not have the red color from the female, does not have the white color from the male, but they blend their characteristics so that they can all exist equally in the phenotype. So it is known as codominance. Now, codominance also exists when we look at blood groups, because in our blood there are 4 types of blood groups. There's blood group A, blood group B, blood group AB, blood group O. This is a response to blood producing certain type of antigens when they are attacked by a certain infection. So the type of antigens that they produce make them be labeled as blood group A or blood group B, blood group AB or blood group O, and blood group O does not produce any type of antigen. So the alleles that represent the different blood groups can be shown here by these letters here. This one is for allele for blood group A, this one is an allele for blood group B, and this one is supposed to have small caps and it represents the O blood group and it's the recessive one, whilst these both of them are going to be dominant alleles. So when fertilization occurs, we are going to see different combination of these blood groups combining to form the different types of blood that we see. So why is it necessary for us to study this? Because if a person has blood group type A, if they are exposed to blood group type B, blood group type B will be a foreign object, which then they need to respond to and destroy. So that's the reason why when there's a blood transfusion, a blood type A is not transfused with a blood type B. A blood type AB now can respond by producing antigens from either blood type A or blood type B. So people with blood group AB can be given either blood types because they are not going to be affected by any of them being a problem as registering as foreign bodies. But then if you have blood group O, you do not have the ability to produce those antigens. So in emergency times, you only need blood group O. You cannot be transfused with blood group AB or blood group A or blood group B. That's the problem with blood group O's. Therefore, if you cross a blood group A person with a blood group B person, the expected results can be as follows. So this is a typical crossing between a person with blood group A and a person with blood group B. If you look at them, both of them are heterozygous. In the sense that this represents the genotype for blood group A, and this represents the genotype for blood group B. Segregation during formation of gametes will result in the formation of these typical gametes, and if you cross them, they will produce these following genotypes: one genotype representing blood group AB, one genotype representing blood group O, one genotype representing blood group B, and one genotype representing blood group A. That is after typical monohybrid crossing occurs. Now, from there, let's look at sex chromosomes. Within our bodies, sex is also determined by chromosomes. And the sexual chromosomes are labeled the X chromosome and the Y chromosome. So the existence of a homozygous or a heterozygous pair of these chromosomes will give us a certain outcome. If the pair of chromosome is two X chromosomes, the resultant phenotype is going to be a female. But if it is an XY chromosome, the resultant phenotype is going to be a male. So if we separate the chromosome at gamete formation, one will have X and the other one will have Y. Or in the case of females, one will have X and the other one will have X. So the resultant crossing of these chromosomes will give us a 50% chance of either the female or the male be produced when fertilization occurred. Right. How can we show this? Genetic crossing. As you can see from this diagram, when a male crosses with a female, the genotype that we expect from the female is an XX, and the genotype that we expect from a male is an XY. By doing these crosses, you can see that the resultant F1 generation is two of them being represented by a homozygous XX, and then two of them being represented by an XY. So in actual sense, we are saying of a possible 4 children, 2 of them are going to be females and 2 of them are going to be males. That's why the phenotypic ratio of the monohybrid crossing for possibly crossing of a male and a female is 1 male is to 1 female. Thank you.